Welcome to Vital Biology classes. For new updates, subscribe to the channel Vital Biology. The theme of our channel is Study Now, Be Proud Later. You can join our Vital Biology Telegram group and all the classes, notes, PDFs are available there. Link is provided below in description box. Hello everyone, welcome to the Vital Biology classes. This class is for all the second PU biology students and it's also helpful for all the NEET and KCET aspirants because biology is also included in this exams. And one thing, before starting this chapter 2, new chapter, that is sexual reproduction in flowering plants, let's revise some words that in chapter 1. Okay, that is like what is reproduction, lifespan, and asexual types of reproduction, that is asexual reproduction, sexual reproduction definitions, and asexual reproduction examples. And in the sexual reproduction there are 3 events, that is pre-fertilization event, fertilization event, and post-fertilization events. So these all we have studied in the previous classes. Okay, who have not seen that classes, first go through that classes and then you can understand this classes, because the basic words like what is sexual reproduction, asexual reproduction, this type of words are clearly and with the definitions and with examples I have explained in the chapter 1, that is reproduction in organisms. Okay, then after listening to that classes, just go through this class, then you can understand it clearly. Okay, so today let us go through this chapter, that is sexual reproduction in flowering plants. Oh, here in this chapter let us study only about the flowering plants, that is sexual reproduction. Okay, so as you know, all love the flowers, right? So flowering flowers are the very different and attractive and interesting thing, living thing, that is very loved by everyone. So these flowers are very helpful in the manufacturing of the scents and perfumes. Okay, these flowers are very helpful in the manufacturing of the scents and perfumes because there is a very different types of odors, smells. Okay, and these flowers shows the sexual reproduction. Flowering plants all shows the sexual reproduction, and here these flowers are very rich in their color and odor, right? It is very attractive to everyone. And these flowers are not only for the human selfishness, it is also helpful in the orientation purpose and ornamental purpose, and socially when any great persons have got their death means, so that for their respect we will keep the flowers, right? In that way, and religious and culturally cultural values, like in temples we will use, and in marriages also these flowers are very useful. And also it is symbols for conveying the important human feelings such as love, affection, and happiness towards the another loved person. Okay, so that you can observe here one thing, like these have the different in their structures. You can observe here they are morphologically and structurally different to each other. You can observe this flower is not similar to this flower, right? And this is not similar to this flower. So everyone has its own morphological and structural characters and their own odor. Okay, this is some importance of flowers. Okay, so now let us move on to the next, that is have you ever seen this flower in your childhood in 8th, 9th, 10th? Yes, obviously you will be seen, because this is the LS of hibiscus flower. So in hibiscus flower, what and all it comes, like stamen and calyx, that is outer layer after the petals. Okay, so petals, this is a very important and attractive part in the flower, that is very colorful in every flower. It is not similar in all the flowers, it is different in every flowers. Here it may be pink, and in previous slide I have showed, here in this flower you can observe these are the petals. So here this is yellow color, and here this is orange in color, and here this is pink, here blue, purple. In this way these are the petals. Okay, and next after the petals, let us see this another part, that is like stigma. So the stigma you can observe here, this is a stigma, and in that the filament is like you can observe very thin in structure and it is very sensitive, that is style. And ovary, so this stigma, style and ovary, totally it is called as pistil. Okay, this pistil is a female reproductive structure in the flowering plants. And the next, that is anther, this yellowish powdery like structure, that is anther. And below the anther, if this is a anther, below the anther there is a thread like structure, this is called as filament and this is called as anther. So totally this anther and the filament is called as stamen. So the stamen is a male reproductive organ in the flowering plants, and this pistil is a female reproductive organ in the female flowers. Okay, I think you are understanding. Let us move on to the next one. Sorry, one second. That is main topic. Okay, so here let us study about the pre-fertilization. As you know, in the last chapter, as I told you, just go through that chapter first and then come to this chapter, because there also there is a in brief way I have explained what is pre-fertilization event and fertilization event and post-fertilization event. So now today let us see here that pre-fertilization. So first let us go through one definition of it, and next we can move on to the next. So what is pre-fertilization means? The formation of the gametes before the fertilization is called as pre-fertilization. Means like before the fusion of the male and female gametes, there is the formation of the gametes, means the preparation of the gametes, that is called as pre-fertilization. Means preparing for the fertilization, that is pre-preparing. Okay, then fertilization, then post fertilization. So here before fertilization called as pre-fertilization, means the formation of the gametes, means it is preparing for the fusion of the male and female gametes. And next, several hormonal and structural changes are the initiated, which leads to the development of the flowers. So several in the human, you can see before the human matures there are several structural and hormonal change, right? After the hormonal and structural change itself, the human matures. It may be females or males. In the same way, here in flowers also, there are several hormonal and the structural changes that starts for the development of flower. If the flower is developing, then it is, then that means it is starting for the fertilization, means for the reproduction. Okay, so this is the thing that occurs before the fertilization, means hormonal change and structural change, that is the starting point for the reproduction. Okay, and the next, inflorescence are formed which bears the buds and then flowers. Inflorescence, it is nothing but group of buds or group of flowers. So next, after this hormonal and structural changes, there is a group of flowering and buds. There is a group of buds, and then these buds itself then leads to the flowering. Okay, blooms. And next point, let us see that, sorry, one second. Yeah, that is androecium. So one second, that is androecium. So what is androecium? Androecium is named for the male reproductive structure, that is, as I showed you below itself, the stamen. I showed you, right, stamen. So this stamen itself, it is called as androecium. Okay, androecium. So this androecium is a male reproductive structure. Okay, male reproductive structure in flower is called as androecium. And next, gynoecium. Gynoecium is a female reproductive structure in the flowers. Okay, in flowers the female reproductive structure is called as gynoecium, and it is also called as, in the common way, it is called as pistils. I have showed you in the hibiscus plant itself. Okay, so next let us move on to the next one. That is, first let us take the stamen. As I told you, it is a male reproductive structure seen in the flowering plants, that is stamen and microsporangium and pollen grains. These three also, all the three also include in the male reproductive structures itself. So first let us see the stamen. Okay, So this is a stamen. So the structure of the stamen you can observe here. This is a structure where this part is called as anther. Okay, remember this and keep that in mind. So this yellow color part is anther and the elongated part is filament. Okay, this is a diagram. Now let us see the theory of this points. Okay, so the long and slender stalk like structure called as filament, as I told you. So this is a long structure that is slender and stalk like a stick like structure, that is called as filament. Okay, that is called as filament. And next, terminally means upwards. Okay, upwards there is a bilobed, one second. Okay, you can observe here terminally bilobed structure called as anther. Okay, so here you can observe that this is the upper part, right? So this is the upper part, and so this is terminally bilobed structure. You can observe here that there are two bilobed like structures in this way. Okay, so this is a anther. Okay, so let us move on to the next point, that is proximal end of the filament attached to the thallus or the petal of the flower. So as I told you, petals I showed you previously. So one second, I'll erase it. So the end means this end part is attached to the thalamus or else petals of the flower. So here the petals will be, right? So in this way the petals are present. So here you can observe the end of the structure, that is proximal end of the filament present at the petals. Okay, so this is the thing. And let us move on to the next point, that is number and length of stamen are variable in flowers of different species. So all the stamen structure will not, all the stamen structures will not be in this same size and number and length. Okay, some stamens may be in a short way, only this much. Okay, and some may be very long than this stamen. Okay, in this way in different species this stamen's structure varies in length and number. Okay, let us move on to the next slide, that is about anther. So still now we studied about this thing, right? So this much we studied. So now let us go through only this part, yellow color part. Okay, that is bilobed structure called as anther. So anther is a, as I told you, anther is a bilobed structure, each lobe having two theca, having two theca called as dithecous. Okay, so this is one lobe. Okay, one second. This is one lobe, you can observe here, and this is another lobe, right? So when we cut this into LS structure, crossly if we cut, you can observe this type of structure. Okay, inside, okay, in this inside you can observe this type of structure that consists, as I told you, bilobe na. So in each lobe means this is one lobe, right? So here you can observe this is one lobe and this is another lobe. In each lobe there are two theca, so that is called as dithecous. So this is one theca and this is another theca, so this are two in each lobe, right? So it is called as, two means nothing but di, right? So dithecous. Okay, each lobe has two theca called as dithecous. Okay, and in another lobe there are another two theca called as dithecous. Okay, so this is some information, and let us move on to the next point, that is like often a longitudinal groove run by separating the theca. Okay, often means later this longitudinal groove here you can observe here somewhat lined structures, right? So this longitudinal grooves just separates, means like it is together, right? These are jointed, so it just separates, means in this way means it will not be jointed, it will just be separated in this way. Okay, it will just separate. Okay, first it was jointed and later it takes place in this way. This longitudinal grooves that runs and separates the theca. And okay, I think this have been understood. So let us move on to the next point, that is the anther is of 4 sides. As you are observing here itself, you can observe here there are 4 corners. So this is one corner and this is one corner and here. So 4 corners are there. As like your home consists of 4 corners, right, in room. So in that way there are 4 corners. That is called as tetragonal structure. Tetra nothing but 4. Okay, so tetragonal structure, it is called as tetragonal structure. And tetragonal structure that consists of 4 microsporangia. Here you can see that this orange color structures right here. So those are the microsporangia. So here there are 4 microsporangia as you are observing here. Orange color are there 4, no? So these are the 4 microsporangia in the corners. In the corners it is there. And 2 in each lobe. In each lobe there is the 2 microsporangia, right? There are only 2 lobes, right? So here also you can observe here in each lobe there is 2 microsporangia. I think you are getting it. If there is any doubt, please ask me in the comment box, okay, so that I can explain you in the comment box in the YouTube or else in the Telegram channel itself. Okay, so let us go to the next point. That is next, microsporangia further develops and becomes a pollen sac. So this microsporangia itself means this orange color part itself, it will develop further, little bit it will develop and further it converts and it becomes to the pollen sac, or else it's also called as pollen bag. Okay, it is also called as pollen bag, where if for example if you take if you purchase or if you shop if you purchase some sarees, you will keep in saree box, right? In that way the number of pollen grains that is carried are collected in one pollen sac or else pollen bag. Okay, in this pollen bags, then it contains these for example let us take this itself here. So these are the 4 pollen bags it consists. Okay, in this pollen bags further there are several pollen grains which contains the several pollen grains. These pollen grains are more than 1000 in number. Okay, I think you have understood this slide. So let us move on to the next slide. That is about structure of microsporangium. So here you can observe that this is the structure of microsporangium, that is viewed normally, but this is enlarged structure. So this part is enlarged structure and this is just seen. Okay, so this is a structure of microsporangium. Okay, so let us see about the structure of microsporangium. Here this microsporangium appears nearly circular in outline. So here you can observe these are almost circular in structures, right? So these are the 4 somewhat like circular in structures. One second, can you see this here? These are the microsporangiums. Okay, these are structural, right? Circular in structure, right? These are circular in structure. So this circular structure means a microsporangium that consists of 4 wall layers. So you can observe here they consist of 4 wall layers, right? So let us observe that this is a enlarged structure. As I told you, this is a enlarged structure. So now let us see what are those 4 layers. So first layer is epidermis. It is the outermost layer, and the second layer is endothecium. Here you can see endothecium. So third layer is middle layer, named as middle layer, and the innermost layer is tapitum. You can observe here this these cells are tapitum. Okay, So now let us see some more explanation about this layers. Okay, so as I showed you in the below slide, you observe that which are the layers, right? That is epidermis, endothecium, and epidermis, endothecium, middle layer, and tapetum, right? So in that, the epidermis, starting 3 layers, that is epidermis, endothecium, and middle layer, are the outermost 3 layers which protects the pollen grains. Okay, which protects the pollen grains, as I explained you. So the pollen grains are present inside this, right? In this microsporangium, right? So in these pollen grains are protected by, as you are observing here, these pollen grains are protected by these layers, right? Okay, this one layer, and the second layer you can observe here, third layer, and tapetum. Okay, this pollen grains are supported by, or else protected by, these 4 layers. Okay, in that, the outermost 3 layers are epidermis, endothecium, and the middle layer. Okay, and it functions in the protection and helps in the dehiscence, means the breaking of the layer. Okay, breaking of the layer and release of the anther, and it helps in the release of the pollens. Okay, inside there is the presence of pollen, right? Here also you can observe, here are some of the pollens, here also pollens. These pollens are then, after the maturation of the microsporangium, these are released out. Okay, these all pollens are released outside in this way. Okay, so it helps in this releasing of the pollens. Okay, next, the innermost layer, that is tapetum. Tapetum is one of the innermost layer that helps to that it nourishes the development of the pollen grains. It helps to nourish the pollen grains. Okay, next, cell of tapetum possess the dense cytoplasm. So the tapetum cell, here you observe that, right? So let us take one of the tapetum. So this nucleus containing tapetum, all are these all are the cells of the tapetum. So let us take one of the cell. Here there is no need. Here let us take. So let us take one of the tapetum cell. So tapetum has a dense cytoplasm, means it has a much fluid content inside the cell, and it also consists of more than one nucleus, means inside it consists more than one nucleus. It may be 2 or 3, 4, anything, but more than one. Okay, so next point, that is sporogenous tissue occupies the center of each microsporangium. So inside this, again I'll go there, right? So so inside this you can observe here, inside the tapetum layer, after the tapetum there is a sporogenous tissues. So here you can observe the empty sporogenous tissues, right, without nucleus. Here you can observe in tapetum there is a nucleus, but in the sporogenous tissue there is no nucleus. Okay, so these are the sporogenous tissues, this empty inside the tapetum. Okay, okay, so that is that sporogenous tissue is present inside or center in the microsporangium. So let us move to the next one, that is microsporogenesis, means the process of formation of pollen grains, or else, yeah, that is the thing, the process of formation of pollen grains. So let us see here the definition of the microsporogenesis. The process of formation of microspore from the pollen mother cell or else pollen grains. Okay, the formation of the pollen, the formation of the microspores from the pollen mother cell is called as microsporogenesis. That is with the cell division, that is meiosis. Okay, here you can observe that. So here they are taking one of the cell, that is this, then it is forming into 2 pollen grains. Okay, 2 pollens, means pollen mother cell, anything, it's okay. So by the cell division, that is meiosis 1. This meiosis and the mitosis you have already studied in the first video. So after the two division, two cell formation, next my meiosis 2 takes place. So here you can observe, I have mentioned you, here you can observe that meiosis 1 gives 2 pollen grains, and next meiosis 2 that gives 4 pollen grains. Again this 4 pollen grains are the microspores, just separates, and this pollen grain matures. Okay, matures and gives a matured, like it releases the pollen grains. Okay, this much. So now let us see the theory. Okay, as I told you, the process of formation of microspores or else pollen grains from the pollen mother cell through the meiosis cell division is called as microsporogenesis. Please practice this definition because it's a very important thing. Next, microspores forms and arranges the cluster of 4 cells, that is called as microspore tetrad. So these are the microspore tetrads. Okay, tetrad is nothing but 4 cells. Okay, next, as anther matures and dehydrates, the microspores, sorry here there is a mistake, it's not a F, R. Okay, as the anther mature and dehydrates, the microspores dissociate from each other and develops into the pollen grains. As I told you, after the maturation of microspore, after this maturation, it forms a matured pollen grains. Okay, it develops into a pollen grain and it just matures. Okay, then there is a thousands of pollen grains released out of the dehiscent anther. Then here this gets separated, this layer just breaks down, and this pollen grains comes out and forms a thousands of and releases the thousands of pollen grains. Okay, so let us move on to the next slide, that is about pollen grains. That is about pollen grains. So pollen grain represents the male gametophyte. As I told you in the beginning itself, the pollen grains is the male gametophyte. Okay, it belongs to the male reproductive organ and it is a male gametophyte. And one thing you can observe here, there are variety of architecture, means variety in pollen grains. So here you can observe that this is one type of pollen grain and this is one type. There are several various types of pollen grains, and in the different styles and different sizes and shapes and different in colors too. Okay, different in colors, designs, everything. So in different species there are different type of pollen grains. Okay, and these pollen grains are generally spherical in shape. You can observe here almost all the pollen grains are spherical in shape, right? So you can observe here, sorry, one second. Can observe here these all are somewhat round in shape, almost all are round in shape, some are elongated. So some are elongated and except some, almost all are spherical in shape, and its size, it's also almost 25 to 50 micrometers in diameter. Its size is up to 25 to 50 micrometer in diameter. Okay, so let us move on to the next slide, that is the pollen grain. Okay, still now I told that the shape is different in different species and size, style, colors and all different in different species I told. So now let us see the deep structure, means like expanded structure, that is the pollen grain is of two layered walls. The pollen grain consists of 2 walls, that is exine and intine. So I have mentioned here exine and intine. So the first layer is the exine. Okay, that is outermost layer, hard outermost layer, that is exine. In pollen grain there are 2 layers, that is exine and intine. The exine is the hard outermost layer. So this dark green color, that is exine. Okay, that is made up of sporopollenin. Okay, this is made up of sporopollenin, means that exine consists of sporopollenin. Exine has prominent aperture called as germ pores. So you can observe here in exine, so here there is somewhat pore, means hole like structure, that is called as germ pore. Okay, so next let us go to the inner wall of the pollen grain. So here you can observe second layer, first one is dark green, right, and the second one is light green, that is the inner layer of the pollen grain, that is called as intine. Okay, that is called as intine. And intine is a thin and continuous layer that is made up of cellulose and pectin. So this intine, innermost layer, that consists of that is made up of like with the cellulose and pectin proteins, and these are continuous and this is thin. Intine is thin layer when compared to the exine, that is outermost layer. Okay, so next point, that is the cytoplasm of the pollen grain is surrounded by the plasma membrane. You can see that. So this another line, okay, you can observe here, this is the another line that is called as plasma membrane. So cytoplasm is covered by the plasma membrane. Okay, this is this fluid like empty structure, this everything as cytoplasm, and these are the vacuoles. Okay, this somewhat white shaded like color, that is vacuoles. Okay, and this red colored structure, that is vegetative nucleus, and this is germinative generative nucleus. I'll be explaining that in the next slide. Okay, so let us move on to the next. When the pollen grains is matured, after the maturation of the pollen grains, it contains two cells. Okay, after the maturation, before maturation there is no any cell content, but after the maturation of the pollen grain there are two they contain two cells, that is vegetative cell and the generative cell. So first let us see what is vegetative cell. So vegetative cell is a bigger, vegetative cell is a bigger, you can observe here. One second. So this is a vegetative cell. Okay, so here you can observe this is a vegetative cell, this one. Okay, so this vegetative cell is bigger in structure and it stores the food, it stores meal, and it is large and irregularly shaped nucleus. It consists of large and irregular shaped nucleus. Okay, so let us move on to the next, that is germinative cell. So germinative cell is smaller when compared to the vegetative cell, and this generative cell is a small in structure when compared to the vegetative cell, and this generative cell is floating in the cytoplasm of the vegetative cell. Okay, and it is spindle in shape and densely cytoplasm, and dense cytoplasm means consists of more cytoplasm fluid structure, and it also consists of nucleus. So you can observe in much of the plant the generative cell that is binucleated. In almost all the plants, in almost all the flowering plants, the generative cell that consists of binucleated. Okay, two nucleus only it consists, but in some of the plants the trinucleated generative cell is also seen, but much plants, flowering plants, that consist of binucleated itself. Okay, so next let us see some more importance of the pollen grains. Okay, so like pollen grains, they are rich in nutrients. Okay, these pollen grains are rich in nutrients, means in carbohydrates, proteins and all. Okay, and nowadays it is a fashion in recent years, like pollen tablets are used. So here you can observe there are some tablets, tonics and syrups, and some powdery like and seeds like structures, right? So these are the pollen seeds stored, and pollen tablets are used for the food supplement. Okay, because by the pollen grains there is also some nutrients are present, where it is used by the humans like pollen tablets. Okay, like in western countries, okay, large number of pollen products like tablets and syrups are available in the market. Okay, and here another one thing, so many of the pollen grains like they live only for 30 minutes. Okay, after it the pollen is transferred. Okay, and some plants, that some flowering plants, that it may live for months ago. Okay, so this much is about today's class. So let us see in the another class. So thank you so much. Bye bye. Thank you. all the class notes and other study materials provided in our Vital Biology Telegram group. Link is provided below in the description box. Like, share, subscribe and press on the bell button for the notification of newly updated classes.